Sairam, welcome to the fundamental unit of life. What's the fundamental unit of life? Yeah, cell. Cell is the fundamental unit of life where it decides what is it and how is it, okay, and how it works. Let's see the interesting part of the cell. So how it works, what are the parts of the cell? Well, we divide this class into three parts. So in part 1 you learn all the definition and who found out, okay, and what are types, you learn that. The rest will fall in part 2 and part 3. So let's begin. Good to go? Whoa, you see something on the board over here? You see two types of cell. If you see here, you have this cell here. This decides how the equipment works. This is the cell which is used for the equipment. Okay, so here it's coming down and then again recharging it. When you recharge it, it goes up. So here you go. It recharges and goes up again. And the same cell which is used everywhere in other equipments is used in our body and all the living beings. That is the cell. Okay, you can see the cell which is functioning. It has got lots of organelles. Indeed, we'll study all the organelles. It's a very interesting thing, and you'll be studying all the organelles together. Here, this gives energy to all the components, but here it gives energy to all the living things. All right. So let's go to the next one. What does it define? It is the smallest structural and functional unit and of an organism which typically it's microscopic, consisting of cytoplasm and a nucleus. It's the most smallest thing. Well, if you say, no, I'll see my cell, if you could, yes, you can see, but not the details. All right. But here, if you see, it is the smallest and structural function unit of an organism. There, it sees how it looks like. All right. It's very microscopic. What is microscopic now? Something to do with microscope? Well, yes, this is a very, very interesting thing. We will be studying the types of the microscopes here. You will be studying how to operate a microscope. Okay, you will be studying how a microscope works. Isn't it very interesting? Yes, it's very interesting. All right, and here you have this. It is enclosed in cytoplasm, nucleus closed in membrane. You can see this is a membrane. Okay, what's a membrane? This is a covering. You will see. You learn the types of membranes, you learn cell types, what it is, how it works like, okay? You will see it in the next coming classes. Now, if you go to see how does a cell evolve, okay? Now, you have this cell. From cell, you can make so many else things. As you take, in case of this equipments, a small cell, you group it together, you have these batteries. Isn't it not? Yes. Now here, also if you take this cell, you have so many things. Can you guess what will come here? Yes. You have this tissue. Now tissue is nothing but group of cells working together. That's called as a tissue. And you'll hear it so many things. Tissues are there in our body, in all the living organisms. Alright? And what's next after tissue? Guess, come on. Open up your minds. Open up, think up. Never mind if it's wrong. Yeah. Here, it's an organ. So all these tissues come together and form an organ. What's an organ? Yes, organ is something which, makes work, all the functions works, everything works there. And what does this form? Yeah, you're right, it's organ system. Correct. So organ system is nothing but group of these organs forming together, working together. It's an organ system. And here, yes, the organism. So, see how cell is important. If cell is not there, all these things cannot be there, right? So, you have this organism. Organism is made up of all of these things, right? So, here, it can be compared to an organization. You go to an organization, all right, as the head, okay? A small one that contributes to so many departments, only all these things together. If that is working, everything works together. The same here. The small thing which is here that contributes to so many things. Alright, every minute, it's like drop by drop fills the mighty ocean. Drop by drop cell fills the mighty organism. Isn't it not? Fine. So let's go to the next one. Have you ever wondered what is this unicellular and multicellular? Uni one, multi many, right? Unicellular is something which is only one cell. Multicellular is something which is more than one cell. Yes, more than one cell. And let's check out the differences between unicellular and the multicellular. Here, unicellular organisms are composed of a single cell, as I told you, and multicellular organisms are composed for more than one cell. And unicellular organisms are irregular in shape, and multicellular have a definite shape. They have got a definite shape. And here you have this is irregular in shape, somehow amoeba, it's somehow in shape, have a definite shape. Multicellular organs, everything, they have a definite shape. Alright, it has a simple body organization. You take this amoeba only, very simple, however it is, but there it's a complex body or organism, multicellular. So if you take this organs and they have complex everything, so many clubbed together, then you have that. Alright, here a single cell carries out all the necessary life processes. So again in the amoeba, it carries out all the necessary life process which is there. But here, multiple cells perform different functions. Multiple, different cells. So for the example, you have this organ, so many cells are there. They perform different. For instance, you have this heart cell that performs different. Okay, kidney cell that performs different. So different complex things. All right. The total cell body is exposed to the environment. All right. You have this. It's there. Amoeba is all over here. It comes into the air, and then afterwards it comes to the water. Then when you consume it, it starts working. All right. Thus, the outer cells are exposed to the environment. Only the outer cells. Inner cells, everything complex in there, but the outer cells are exposed to the environment. Here, they are microscopic in nature. They can be seen only under a microscope. They are microscopic. Yes, little bit bigger. They can be seen by the naked eyes. All right. And then examples for this are this bacteria, amoeba, paramecium, yeast. All of them are unicellular organisms. But in multicellular, you have this human, animals, plants, birds, insects. All of them are multicellular. Got the concept correct? Yes. So this is the difference between unicellular and multicellular. Uni one, remember, uni one, multi many. Alright? So you have this. Now, before going to all these things, somebody leads to some discovery, and then voila, you have new things. So let's study who discovered the cells, how did it come into effect, okay? And afterwards we'll study the microscope. So cell, all right? So here, can you guess how it was discovered? Yeah. So what happened in 1665, Robert Hooke, he was an English scientist. He saw that he discovered this cells, all right? And then he saw a thin slice of cork with his crude microscope. So crude, you'll see this is a crude microscope. So what he did, he saw this small cells in a thin slice of cork. He took the cork, he scraped it. So when he scraped it, what happened? He got some small things. So he thought, let me see under my crude microscope, the first microscope, let me see how it looks like. So when he saw under that, he saw something like honeycomb cells. You've seen the honeycomb? Yeah. If you see the honeycomb, you see structures like this, all right? Something like this, you see like this. So it was just similar like this. That's the honeycomb structure, all right? And he termed them as cellulae or cells. So that is how he termed that. And who was it? It was Robert Hooke. Remember this word, Robert Hooke. It's a very important thing, all right? And then his discovery indicated first time that living organisms consisted of number of small structural units. So can you see the small, so many structural units which are here? So first time his discovery indicated that this living organisms consisted of small structural units. All right. So that is how the cell discovery started. They discovered. Now one question to you: what's the difference between discovery and invention? Think. Yes. Discovery means it is still there, you take and you just come out with it. Invention is coming out with a completely new stuff. Okay, like you have this invented the computers, but you discover the cells because cells are already there. You not invent them. All right. So here you know Robert Hooke discovered this cells. So this is how the cells look like. Now again continuing with the history of cell. So if you go to see the history, something is done and many people put light on that and they go forward, they go on advancing forward. For example, if I find out something in the microbial fuel cells and I say that okay, this one leads to this one, by using this fuel cells you can run electricity. I use one step, I use waste things and I run. And but another person comes and he thinks waste things here, but I can add some more things and go forward to find out how it works, and that's how it led to cell theory and other things. So what did they do? They went on further to discover what are the cells, how are the types, what should be there in that. So all these things was discovered here. Alright. So if you see 1665, Robert Hooke discovered the cell. See, Robert Hooke has here, right. Then in 1674, Antony von Leeuwenhoek studied the living things for the first time, and he's the one who founded this microscope. Alright. So this Leeuwenhoek is the one, alright, who studied the living things for the first time. Studied in the sense, okay, studied in the sense he made it sure what does it look like, how does it look, okay, and how does it do. So all these things he studied. Alright. And then you have this 1831 Robert Brown. He discovered the nucleus. So this Robert Brown discovered the nucleus. You'll come to know what is this nucleus, what is the cytoplasm, okay, what this comes under cell organ. You'll be studying in the further classes. Alright. And then in 1838 to 39, M.J. Skleden and Schwann formulated the cell theory. So Skleden and Schwann, they formulated the cell theory. Now cell theory means they came up with a theory, okay, cell looks like this, yes, cell consists of all these things and blah blah blah, all these things are there in that and we need to use this is how a cell is, we have this, all these things were there in this, and he formulated the cell theory. In 1855, you see this Rudolf Virchow, okay, this Rudolf Virchow stated omnis cellula hi cellula. He gave a term to the cell. He stated that omnis cellula hi cellula means cell. It's a unit, single basic unit, okay? It doesn't depend on, it is like energy center. So he termed this omnis cellula hi cellula. So these are the scientists, who worked on this history of the cell, okay, discovery, and then doing so many other researches and so many things, and that's how we learn the cell today. A very interesting topic to go, okay, and that's how we learn cell, and it goes, okay, because of the cell we are alive today, or else if there's no cell we're not alive. So as you have that battery cell, if the battery cell is alive, you have all this equipments running, right? The same thing you have your cell, okay? If you have the cell in us and everywhere, it keeps us alive, alright? So let's go forward in this. So if you see the microscopes, okay, these are the types of this microscopes, alright? So see, Leeuwenhoek microscope, alright, in the 1600s. So what he did, this is the microscope which he, invented. This is the first microscope, which is after the crude microscope, he invented this microscope. Can you guess what is the size of this microscope? Yeah, come on, guess, it's okay, no problem. Yeah, it is just the size of your thumb, that's all. Okay, it was just the size of thumb, and people used to go and used to observe and see what it is, how it is. Yes, and that's how, this Leeuwenhoek founded this, and he saw the cells, he analyzed. Imagine, he saw everything. Nowadays you have this complex microscope which can do just in the snap of a finger, you'll get everything, okay? All these things, all these images and everything, you'll get just a snap of a finger, okay? That's because of the advanced microscope. Now coming to the type of this microscopes, okay? This is a simple microscope what we have here. Then you see something here which is just looks like a satellite or something. Yeah, it's not a satellite, it's a dissecting microscope, okay? Here, this microscope is used to dissect the plants, okay? You take this plant cell, you take this, okay? You observe it, you take this tomato, you observe everything. Observing, that can be done using this, alright? a simple microscope or simple, it's called as a dissection microscope. Very simple. You take this, okay, there's a, eyepiece, you take this, you just put on this stage, you put a specimen on the stage, and then you can observe it, and that's how it will show the enlarged view. It'll enlarge it. Basically you have 10x and 20x, okay, which enlarges it 10 times and 20 times. And then coming to this microscope, can you guess the name of this? Yes, it's called, you're right, it's called as compound microscope, right? Compound. Why compound? Because things are compound and complex there. It magnifies it much more times than this one, okay? And it has got so many things here. See, you've got this ocular lens, which is eyepiece, you've got the body tube, you've got the revolving nose piece, you've got the arm, you've got the stage, you've got the stage clips, you've got the coarse adjustment knob, you've got the fine adjustment knob, you've got the diaphragm here, you've got the light source, you've got the base. Well, we'll study this in detail. Alright, so this is what all these parts are in this compound microscope. Can you guess what is this? Well, no, that's not a computer. Yes, you're right. It is an electron microscope. Okay, the two types of electron microscope: transition electron microscope and scanning electron microscope. Now you must be wondering, what is this electron microscope? Well, electron microscope, the name itself suggests there, right? It is a microscope which magnifies this thing at the electron level. Even the electrons are seen. Yes, the hot topic, the coronavirus, which you see, the image which you saw of the coronavirus, the round thing and those sticks here there, that's got by this microscope. Which one? Yes, the electron microscope. That's got by this electron microscope only, okay? And it requires a lot of things to magnify it to the smallest level. All the images of the bacteria, okay, the paramecium, all these things which you see are magnified by this. Yes, this microscope magnifies it to the smallest level, to the nanometer level, decimeter level. So very smallest things can be seen by this microscope. Alright, here you just see it. It is just in 10 times or little bit. Here 10 times it is magnified. Here 100 times. Here 1000 times. Alright, isn't it more powerful? Yes. All the research work, complex research work regarding cells are carried out by both of these microscopes: scanning electron microscope and transition electron microscopes. So here you have this, this microscope. If you see here, alright, it magnifies it to the electron level. So electron, you imagine electron level means it goes very, very, very deep. You can see all the things in the cell, all these organelles. Basically see all these organelles. How? By this electron microscope, which makes it possible to see everything, right? So let's go forward. So this microscope, now coming from the ancient Greek, micros means small, and scoping means to look. All right, so here microscope, so if you see, it is a Greek term, micros, small. All right, scopin means to see. So basically you see this small things which magnifies it to the n number of times. All right, and this is an instrument used to see objects that are too small to be seen by the naked eye. Now if I say I can see cells, no, you can't see cells, right? So if you take it and you analyze it, you put it into the microscope, you can see it. Alright. So microscopy is a science of investigating small objects and structures using such an instrument. So you take this instrument, so that is a branch of science. They investigate small things. Oh, what is this? What can be going forward? What is there in this? So all these things are investigated by using this. Alright. Microscopic means invisible to the eyes unless aided by the microscope. Microscopic, small thing. Microscopic, which is very, very small, that can be seen, okay, that can be seen only by the microscope. It can't be seen by the eyes. Alright. So here we have the compound microscope. Now to see the compound microscope, here this is called as a compound microscope. Alright. So this is a real compound microscope over here, and I'll explain you all the parts, okay, one by one. All right, there's a basic way to hold this microscope. You can't just hold it like a suitcase and say, I'm going out. No, you can't do it like that. Well, you have to hold it like this. The arm, you hold it like this, and the base, this left hand like this. You need to hold it like this firmly and then take it to wherever you want and put it there and then analyze. All right. So here you have this ocular lens or the eyepiece. Now, why is it called as the eyepiece? Okay. Have you ever been to the watch repair personnel? Yes. If you go to the watch repair person, he does something. What does he do? He takes something and he says it like this and he analyzes, he says, oh yes, some here, and puts it and he does it. Well, that's the eyepiece. Why? He puts it into the eye and he sees it magnifies. Okay. So this is called as the eyepiece of the microscope. So here you can put it, okay, and you see. And here you have something called as a 10x, 20x. It means that x means it magnifies it so many times. So 10x means it magnifies 10 times. 20x means it magnifies to 20 times. Alright. And then you have this body tube over here. You see, this is the body tube, okay, or the draw tube, you can say body tube or the draw tube over here. You have it over here. Alright. And then that draws, this body draws the image. And then you have this arm. This arm, why is it called as the arm? Because you hold it like this firmly. It's the arm of the microscope. Alright. So you call it as the arm. And then you have this nose piece, revolving nose piece. So this is the revolving nose piece. Alright. This nose piece, it revolves. Okay. Can you see it revolving? Yes. It revolves there. So that's why it's called as a revolving nose piece. And then you have this objectives. See, you see something over here. This is the objectives. This also helps in the magnification. Alright, this objectives also magnifies it. It has this lens, okay, which magnifies it. And we have power, like, here also we have this 45x, alright, you have this 10x and so many 100x and so many things you have. That is also done by this, okay. That adds up to the magnifying effect, alright. And then you have this stage. So as you need some stage, suppose you want to perform something, you need some stage, okay. So does the microscope have here to make it visible. You have the stage over here where the slide is been put. Alright. This is a slide. This is called as a slide which is used, and this is put onto the stage. When you put onto the stage, okay, and you can view it. Alright. And you have the stage clips. Can you see the stage clips? So these stage clips are used to hold the slide. Alright. You put it here. The stage clips, if you see, it's a slide is here, and you have the stage clips over here which holds this, alright, which holds this firmly. And then you have this coarse adjustment here. So if you see here, it's the slightly differs from this one and this one. So you have this coarse adjustment. This coarse adjustment, if you see, if you have to see the image properly, you need to adjust it. So at one particular level, you have this adjusted properly. It's same like this binocular. You adjust it and you see the image clearly. So same thing here. What you do, you adjust it over here to see the image clearly. So this is a coarse adjustment which adjusts and sees the image very, very clearly. And then you have this fine adjustment knob. Okay, this is a fine adjustment. After the coarse adjustment, what happens? You have adjusted it, and then there are some of the things which are not visible. So for that you need to adjust it finely. So that's adjusted finely over here. And then you have this diaphragm. Alright, so diaphragm can be compared, okay, this iris, diaphragm or iris, it can be compared to our eyes. So what happens if suddenly there's a lot of light, so you tend to close your eyes like this. But if there's this darkness, you tend to open your eyes a lot, so that a lot of eyes can get in and you can see the image. So same does this diaphragm. It manages the light, the amount of light. Suppose say if you focus on and light is too much, so you can adjust the amount of light coming through the lens, okay. It can adjust it, and then the required amount of that can be adjusted using this diaphragm. Alright. And then you have this light source. So light source can be two things. You can take this mirror, okay, you can reflect the light source and that gets reflected here, and there's one more, there's electrical light source here. So here you have this electrical light source. So you take it from here, the electricity, and there's a bulb here which gives the light source. Alright. So this common thing is this mirror. So the mirror is adjusted, so you get the light source, and then you can see the object. Alright. And then you have this base. Base holds the microscope very firmly. Alright. If you put it onto that, and it makes a stand. It's a stand or the base which which makes it very, very firmly. Alright. So this is the parts of the microscope, and you have, so hope you got the things right. So just to go through the things once again, this is called as the, yes, eyepiece, and this is called as the draw tube, or you can say body tube. Alright. This is the, yes, coarse adjustment. This is the, yes, fine adjustment. Right. And this is the, yes, revolving nose piece. And this is the stage. Good. This is the stage clip. Good. This is the, so what do you call this? Yeah, slide. This is called as a slide. Okay. And then this is the, what do you call this one? Arm. Good. And this is the, what is this? Once again, revolving nose piece. Good. So here, what is this? Yes, this is the diaphragm. And then what is this called as? Good, base. This is called as the light source. What is the light source here? Yes, it's a mirror. What's the light source here? Yeah, it's an electrical light source. So all these things form up the microscope. So this is how the microscope looks like, and it's a very interesting thing. We do use it in our day-to-day life. Okay, if you go to the lab, all these things, they use this compound microscope, and it is used for so many studies. Okay, so coming back to the things. All right. So now when you go, how does the microscope work like exactly? Okay, if you see the lens which I showed you, the lens which are there, okay, these are the convex lenses which are curved glass to make the microscopic. Okay, so this is a convex lens. So convex lens, if you see, put the convex lens here, it magnifies it. Okay, so that is how the microscope works. And then you have this, what do the convex lens do basically? They bend the light and focus it in one spot. You can see it's focusing in one spot. Yes, they bend the light together and they focus it on one spot. Alright, and as I told you, the magnification. Alright, so magnification is nothing but it magnifies it to the number of times. Here, so how do you determine the magnifying? Okay, so here it is written as 10x. Alright, on the objective it will be, suppose you take 40x. Alright, so you have this 10x and objective is 40x. So 10 into 40, it is 400. It means that it magnifies the image 400 times. Yeah. Suppose you put it, you put it another, you say 20, that many times it magnifies it. So that is the power of this microscope. So if you see this is an objective lens. They have this magnification written on that. Alright. So if you multiply it, it is like 400 times larger. So that is how it is. This is the process, and that is how you can change the magnifications also. Okay. Now, as I showed you, this is the correct way to carry out a microscope. So you have seen him. He's holding it like this. This is the correct way to carry out the microscope. Not like just for just swear it around or carry it. You have to carry it with two hands properly. All right. Now let's see the onion peel experiment. This experiment is used, to see how the cells look like. You can do it yourself. All right. So when you take this onion and when you do it, this is how the cells look like. You can see the cells like this, and when you see that on the microscope, you can see this. This is a nucleus. All right. So first, what you do, you take an onion. All right. You take an onion. For that, squeeze the inner layer from that. There's an inner very, very thin layer. You squeeze that layer. All right, and put it onto this. The tweezer. Take the tweezer. Put it onto the slide. All right. You put it onto that slide, and then afterwards what you do, you use something called as a saffron stain. Now you must be wondering, what are these stains? Have you heard about the stains? Oh, I stain my shirt. Oh, I stain my clothes. What is stain? Something which goes there and makes it visible, and it goes and makes it permanent. So saffron stain is one such stain. It is used to view this plant cells. All right. So that is put onto this. Okay, and then hold it off for some time. So when you hold it off for some time, what happens? The cells absorb this stain, And then afterwards what did you take some distilled water, put on top of it, and then flush it up, and then you take this cover slip. This is called as a cover slip, okay, a small slip. So you take the cover slip and put it onto that, and this is what do you call this, pin. So you take this, okay, you take this needle, it's called as a needle, all right. So you take this and then you put it slowly onto that, all right, and with this you just tap it. So your slide is ready. So I'm preparing a virtual slide. I'm not exactly preparing it. So I'm just preparing the virtual slide showing you. So your slide is ready. So when you do this, all right, so you take it and put it into the microscope. Where do you put it? You put it here, the stage, okay, you stay the stage, you put it onto the stage here, fix it here, and then fix it by the clips, okay, and then adjust it, all right, adjust the light source, what I want, whether you should see the light source, and then adjust it to the lightest, you take it to the smallest magnification power, say 10x you take it, and then you adjust the coarse adjustment. Then after some time, you'll see some blurred image. So go little bit and you'll get this image. And you adjust it, finally, you'll get the best image over there. So that is how you adjust the things, and this is the image which you get when you take this onion cells. Alright? And these are the nucleus. You can see the nucleus here. You can see the nucleus, literally see it. You can do it. Alright, you can do it yourself when you have opportunity to see the compound microscope. You just do it. It's very, very simple. You do it. You can see the cells under the microscope. And then now coming to this prokaryotes and eukaryotes. Alright, so prokaryotes and eukaryotes are the cells which were there. Alright, and then you have this prokaryotic cell and the eukaryotic cell here. So this organisms, if you see, the eukaryotic cells are present in the plants, animals, fungi, and all they have this eukaryotic cells. And in this prokaryotic cells, only bacteria and cyanobacteria have this prokaryotic cells, two nucleus. So that's how they have this. And then you have this cell wall. So cell wall is present. It's in animals it's not present. Okay, in plants only it's present in plants. But the prokaryotes have this cell wall. Okay, and if you see the centrioles, yes, all animals and some of the lower plant forms have this. And prokaryotic cell, no, they don't have this centrioles. The cilia and flagella, yes, simple, okay, this eukaryotic cell is there, and complex in case of this prokaryotic cell. So cilia and again in Golgi complex, yes, it's present in the eukaryotic cell but absent, it's not there in the prokaryotic cell. And you have this lysosomes which are common in animals, not present in the plants in case of eukaryotic cells. In the prokaryotic cells, no, it's not there. So peroxisomes, yes, it's there in the eukaryotic cells and not there in the prokaryotic cells. And the nucleus, yes, it's there in the eukaryotic cells and not there in the prokaryotic cells also. And then you have this plasma membrane, yes, it is there, eukaryotic cell also, prokaryotic cell, but it differs from each other. And then you have this chromosomes, several chromosomes are present in the eukaryotic cells, only one long DNA strand is present in the prokaryotic cell here. Alright, and then the ribosomes, yes, they are present in the eukaryotic cell, and then yeah, they are also present in the prokaryotic cell, but they differ a lot. There's a difference between them. And then you have this endoplasmic reticulum also, that is present in the eukaryotic cell and that's absent in the prokaryotic cell. So these are the differences between the prokaryotes and the eukaryotes. So these are the first organisms which are, which have evolved, and this is an important difference which will, it has to be remembered, which will come surely for examination. And then if you see the shape and size of the cell, okay, you see lots of shapes and size, right from the smallest one to the biggest one. All right, so if you see here, you have this sex cell, you have this muscle cell over here, you have this fat cell over here, you have this bone cell, okay, and this from the stem cell only, it's from the stem cell only all this cells comes. All right, you have this blood cell, and you have this nervous cell, all right, and you have this epithelial cell, you have this immune cell. All the cells, okay, they vary from the smallest to the biggest, okay, they vary, like smallest one you have, all right, then the biggest one you have, all right, so they vary from each other. How do they vary? Okay, so you have this nervous cell which it's present in the nerve, it's like kilometers together. It has this all the synapses and everything that is carried out by this nervous cell. This muscle cell is present in your muscles, okay, fat cell is present in the adipocytes in your fats, okay, bone cell is present in your bones, okay, blood cells in your RBCs in your blood, and then you have this epithelial cells which is present everywhere, all right, and then you have this human cell which gets you the immunity. You have the sex cells which is the ovum and the sperm which is present, which is gives life to the offsprings. So all those things are the types of the cells, okay, the shapes and size. So this is how they look like. So if you see, they vary, but all of these are ultimately determine the specific function. So they vary, but they have this specific function. Like it's like this, human beings vary from each person to other person, but they have the specific function, all right. And then you have the some cells can change their shapes like amoeba, WBC, but plants and animals have almost fixed shapes. Only this amoeba can change their shapes, all right. So but plants, all this, they have the fixed shape. And then the smallest cell is PPLO. It means that it's pneumonia-like organisms. Okay, so these are the like these organisms like look like this pneumonia, and they're called as pneumonia-like organisms. And then example is the mycoplasma. And the largest cell is the ostrich egg. That's also a cell. Yes, if you see the ostrich, it's very large. So that's the largest cell. And then the longest animal cell is the nerve cell. As I told you, the nerve cell, right from the top to bottom, okay, head to the toe, this nerve cell is there. If you stretch it, it stretches to kilometers together, okay. That is the longest animal cell, that's a nerve cell, alright. And a micrometer, if you see, it's measured in the micrometer. What is a micrometer? It's a micron, it's a thousandths of a millimeter. So this is how it is, and they play a very important role in our life. All right, and then it is the basic things in our life. Okay, so coming to this end of this part 1, let's see, test your skills. So here, test your skills now. Answer these questions for yourself. Okay, so here, define microscope and name its parts. So as I told you, you can take and you can remember. Okay, name its parts. Who discovered the cells and how? Go to that slide there and see, who discovered and how. All right, and note down the differences between prokaryotic cell and the eukaryotic cell. I give that what's the differences, and then name the largest cell. So the largest cell which is there, just now I told you. Who discovered the microscope? So go back to the microscope and see what it is, and how does the microscope work? How does it work? How does it come into existence? Okay, and then write the process of magnification in the microscope. How does it magnify? Alright, you can do that. Which is the correct way of holding the microscope? I showed you what is the correct way of holding the microscope. Go to that and write this, and describe the process of onion peel experiment. I showed you the onion peel experiment. So go to that and test your skills on that. Close all your books, close everything, okay, and just note on this and test for yourself. Okay, it's always like you test yourself and correct yourself. Okay, and there we end this first part. Thank you. Sai Ram.